type of fat, which is called a phospholipid. And again, phospholipids have a lipid portion to it, which is the two fatty acid chains and the glycerol backbone. And substituting the one fatty acid chain, we have this phosphate containing polar group with a positive and a negative charge. Now I want you to know that a PO4 group is what we refer to as a phosphate group. Okay? We're going to use that terminology, that molecule quite a lot in this class, so just so that you're aware of it. Now phospholipids are only found in our cell membrane. And somebody, when they started drawing phospholipids, they drew this round portion for the polar portion with the positive and negative charges. And so they kind of drew it like this. And then for the fatty acid chains, they drew these two side portions here. And somebody said this looks like a head. And this sort of looks like some tails coming off, and so this stuck. We have a head and we have tails. So the head is the polar region, and the tail is the non-polar region. And these phospholipids, again, they're only found in our cell membrane. So if you're looking at a cell membrane, all right, surrounding this cell is water, basically. And we call this the extracellular fluid. Inside the cell is mainly water, and it's referred to as the intracellular fluid. So this is kind of a problem in what we make the cell membrane with. If we make the cell membrane out of a polar substance, what would happen to the cell membrane? There's water on the outside, water on the inside, and like dissolves like. So what kind of a substance is water? Is it polar or non-polar? Water is polar. So if we make the cell membrane out of a polar substance, it'll dissolve in the water. So you can't make a cell membrane out of something polar because this fluid would just dissolve it away. And what would happen if we made the cell membrane out of something that was purely non-polar? Well, think about what we already talked about when you put oil in water, because oil is totally non-polar. And let's say I drop some olive oil into a container of water and put a lid on it, and then I shake. What happens to my olive oil as I shake the water? It falls apart, doesn't it? It tries to mix, and you get these little tiny bubbles of oil all the way through. So if all of our cell membranes were made out of a completely non-polar substance, what would happen to my cell membrane when I waved at someone? It'd fall apart. My hand would go off and slap them in the head. Literally, your body parts would literally fall apart. Your cell membranes would just come apart. So we can't make our cell membranes out of something polar because they'll dissolve. And if it's totally non-polar, they'll fall apart because they're sitting in this watery environment. So phospholipids are the best of both worlds. They're a little polar, a little bit non-polar. Now, the polar head part, if you're looking at a piece of the cell membrane here, the polar head part, let's say this is the extracellular fluid here, we'll have intracellular fluid here, and our polar heads are going to line up along the edges that have the fluid. And the tails coming off of these heads. And we have a double layer so that the heads again are lining up with the intracellular fluid as well. And the tails are sandwiched in between. Now what allows to happen here is when the water interacts with these heads, it can't dissolve it because they're attached to the tails, so it doesn't have the ability to dissolve. But the water also can't get in between these heads because it repels it away. The heads are super duper tightly packed next to each other. They don't let water in between, because if water could get in between, it would push the tails, which are the oily part, away. And so these heads pack real tight on both sides where we have the polar water and repel the water away so that we don't get dissolving, we don't get spreading apart of the membrane. And we'll talk about this phospholipid layer a little bit later, but it's called a bilayer because there's two layers of these. Okay. By the way, the heads, since they are polar and they do, polar has the ability to dissolve or interact with water. A lot of times you'll hear this called hydrophilic. And the tails, since they cannot interact well with water, these are called hydrophobic. Anytime we have a chemical that is both hydrophilic and hydrophobic, we call this amphipathic. Okay? So amphipathic means that we have this chemical that has a bit of hydrophilic and a bit of hydrophobic to it. The next type of fat-like substance found in the human body is actually steroids. Steroids are a type of hormone. Now we have two different categories of hormones in the human body, two major categories. One are fat hormones, which are the steroid hormones. The second are protein hormones, which we'll talk about in a little bit. Now these steroid hormones, they are initially derived or initially made from cholesterol. And cholesterol is a type of fat. And you can see here, this is the cholesterol molecule. We just change it up a little bit, and we have a steroid hormone called testosterone. If you change this cholesterol molecule a bit more, you have a steroid hormone, which would be estrogen. So we need cholesterol in order to be able to make the steroid hormones. And remember, I told you that people who have very low-fat diets typically do not make many steroid hormones. That's because we don't actually eat a lot of cholesterol in our diet. Really, where we get cholesterol in the body from is from fat. So again, you know that de novo synthesis, we can convert fat, excuse me, into cholesterol. So if a person is actually trying to lower, for whatever dumb reason, the cholesterol levels in their body, they have to lower the fat levels as well. But the best way to lower your cholesterol levels, don't eat as many carbohydrates. Which is true. Alright, so let's talk about the last type of biomolecule. This is protein. Now when we talk about proteins, proteins are basically going to come in two different varieties. One type of protein is called a structural protein. The second type of protein is what's referred to as a functional protein. Now, structural proteins help to make up the form or the shape of the body. So this would be like our bones. Hair is a structural protein. Some connective tissue is structural protein. What else do I have up there? Oh, let's change it. Functional proteins, they have particular jobs that need to be done in the body. So for instance, there's certain proteins that we call enzymes. and we'll talk about these because that's what we're doing in lab today. These are a catalyst. And then we have protein hormones. We just talked about those. Antibodies. Muscles. Let's see what else. Well, there's a whole bunch of them, but that's pretty good for, oh, another one. Hemoglobin. These are some good examples of functional proteins. So before we get into those too much, let's just talk first about the basic structure of any kind of protein. And these proteins are made up of separate chemicals called amino acids. And we're going to take these amino acids and we're going to bond them together to form a protein. Now, the smallest protein in the human body is about 1,500 amino acids long. The larger proteins are about 50,000 amino acids long. So proteins are pretty big molecules, pretty big chemicals in the human body. So when we look at the basic structure of an amino acid, what it has is this NH2 here. This is an amine group. And then the C double bond O, OH, this is a carboxylic acid group. And then it has this R group, or what's also referred to as a functional group. Now there are 20 different types of amino acids we use in our body, and all 20 of them have an amine group and an acid group, which is where we get the term amino acid. But each of these 20 different types of amino acids have a different functional or a different R group. So for instance, glycine, okay? Glycine has a hydrogen for its functional group, whereas aspartic acid has a methyl and a carboxylic acid for its functional group. Cysteine will have a methyl and a sulfur for its functional group. And then the other types of amino acids have a more complex functional group. So each of these functional groups is what makes the amino acid different from the other one. Each of them has an amine group, a carboxylic acid group, and the difference is the R or the functional group. That's it. Now, in order for our body to build a protein, somewhere in that protein, you have to have all 20 of the amino acids. So even if you only have one glycine and then it's made up of all the others, doesn't matter. If you look at a protein, somewhere in its structure, it has all 20 of the amino acids. Now, you and I need to eat amino acids on a daily basis in order to make proteins. But, interestingly enough, we don't have to eat all of them. You don't have to eat all 20 of them. Some of the amino acids we can make through de novo synthesis. Some of them we can't. Some of them we absolutely have to eat every day. Now, this potentially is a problem for humans who are not carnivores. I don't know about you, but I'm a really good, righteous carnivore. I like to eat meat for breakfast, lunch, and dinner, okay? And so I don't have to worry about getting all my amino acid supply because when I eat meat, I'm basically eating all 20 amino acids, okay? But you may have some patients who are vegetarians. And vegetables, certain vegetables, do not have all 20 of the amino acids that we need. So what you have to do for these patients is you have to tell them they need to study how to be a healthy vegetarian. It's really important. Because what they may have to do, not may, they will have to do, is they have to eat certain combinations of food on a daily basis to make sure they get all 20 amino acids. So for instance, if you eat peanut butter on wheat bread, you get all 20 amino acids. If you eat rice and beans, you get all 20 amino acids so that you can build proteins. But if you don't eat even one of the types of amino acids that our body can't make, you can't build any proteins. And this is a problem because think about it this way. Our heart, it beats all day, every day. And as that heart is beating, some of the proteins just naturally break down, they wear out, no big deal. You build them back up again. But what if I'm a vegetarian who doesn't eat all 20 amino acids that I need on a daily basis, and my heart is doing this pumping thing, and proteins are breaking down, but I don't have all 20 amino acids in my diet? Can I build my heart muscle back up? Nope. In the 1960s, they came out with one of the very first types of protein drinks that you could drink to lose weight. And so, you know, this is a big marketing thing, and, you know, mostly women were drinking the protein drinks at that time. And so they had these women all over the United States drinking this protein drink. And about 6 months into this protein drink diet, which, by the way, consisted of eating nothing and only drinking this protein drink for breakfast, lunch, and dinner. Now, of course, I don't know who you are, but if all you do is drink a protein drink for breakfast, lunch, and dinner, you're going to lose weight. Okay? It's probably not very healthy weight loss, but you're going to lose weight. And about 6 months into this, people were dying all over the place. And all of a sudden, somebody noticed, oh shoot, we only put 19 of the 20 amino acids into this protein drink. And the one amino acid that we forgot was one that the human body can't make. Oh crap. So that means these people were eating nothing else except this protein drink, and they weren't getting that one amino acid. And their heart muscle was tearing down until finally it just couldn't work anymore, and people died from it. So this is very important that on a daily basis we get the right combinations of foods to have the appropriate amounts of amino acids so we can build these proteins and do repair that we need every day, all the time. The 9 amino acids that we have to get in our food are called essential amino acids. The 11 amino acids that we can produce through de novo synthesis are called non-essential. So it's essential to eat it, so we have to get it in our food. It may not be, or it's not essential to eat it, non-essential because we can do it through de novo synthesis. Okay, now, when these amino acids are bonded together to form a protein, I want you to know that the bond is called a peptide bond. So when we bond 50,000 amino acids together, we're going to have a lot of peptide bonds, which is why you'll hear people refer to proteins as a polypeptide. Because it has a lot of peptide bonds. Now, proteins can take on various different shapes depending on whether the protein is going to become a structural protein or a functional protein. So the very first shape we're going to see when we start bonding amino acids together is, think about if you could put a bunch of pearls on a string and you just have this straight string of pearls, and each pearl represents an amino acid. So if you could put 50,000 pearls on a string, you would have what we would call a primary protein. This is the primary structure of a protein. Just a long strand of amino acids. Now, this strand of amino acids has the ability to bond to other amino acids. So I'm going to go back to this picture right here. And notice on these amino acids, you've got some oxygens and some hydrogens, sulfurs, nitrogens. These are all coming out in 3D, above, in front, below, behind this long strand of amino acids. And a lot of what's coming out are hydrogens and oxygens and carbons, which just love to bond to each other. And these side structures are able to bond to each other and form hydrogen bonds. By forming these hydrogen bonds, we can actually change the shape of our protein. So what you get now from this long strand, you get these hydrogen bonds interacting, and all of a sudden your protein takes on this kind of twisted shape here, which is what we call an alpha helix. And an alpha helix is a secondary structure. So our secondary structure is occurring due to the hydrogen bonding, and this creates what we call an alpha. The little fish sign stands for alpha helix, kind of like a little spiral staircase thing going on. Now remember, I told you yesterday, I think, hydrogen bonds. They're very weak bonds. Don't take much energy to make them or break them, which also means that they form really fast and they can break really fast. So as soon as this long strand of amino acids occurs, there's potential that instantly the hydrogen bonds begin to form, and you get this alpha helix spiral staircase thing going on, and we call this a secondary structure. Now sometimes what will happen is this secondary structure actually begins to fold in on itself. The ends start to fold over, and you get this sort of funky glob looking thing occurring right here, and the alpha helix that we have has just folded. And any time it folds in on itself and you see this weird looking shape right here, its official term is actually a glob of protein. This is a tertiary structure. Sometimes you'll hear people call this just a glob of protein, or if they want to sound like they're a little bit more educated, you might hear them call it a globular protein. But it's just a weird shape of protein. And this is what we call a tertiary structure. If you take, like in this picture, 4 globs, so there's 1, 2, 3, 4 globs of protein, and put them all together, you have what's called a quaternary structure. So these are the different shapes of proteins that we find in the human body. Any protein that is primary or secondary in shape is always structural. So hair is secondary in shape. Connective tissue can be primary or secondary. Bones have primary and secondary in them. So anytime you have either the long strand of protein or the alpha helix shape, it's going to be some kind of structural protein. Tertiary proteins versus quaternary. Quaternary is always and only hemoglobin. All the other functional proteins, everything else, is globular, tertiary. So protein hormones, antibodies, enzymes, they're all tertiary globs. Hemoglobin is 4 globs together. It's the only quaternary structure in the human body.
So the
Tertiary.
Is it functional?
Yes, it is.
And so is the
Quaternary is functional, but it's only hemoglobin.
so both the third and fourth are functional.
Third and fourth are functional, but fourth is only hemoglobin, and third is all the other functional stuff.
Thank you.
Mm-hmm. Okay, now, hydrogen bonds. They are very weak bonds again. They can form fast, break fast, okay? And we can break hydrogen bonds by adding a phosphate group to them. Now, here's the interesting thing. When we break a hydrogen bond, we can actually change the shape of the protein. So if you add a phosphate group to this glob of protein here, you can change the shape. Because hydrogen bonds create a certain shape, and if I break a few, not a lot, but a couple hydrogen bonds, I can get a totally different shape. So one shape with certain hydrogen bonds occur, and then when I break those hydrogen bonds, I get a second shape. Kind of like transformers. 1 minute it's a car, the next minute it's a robot, right? Okay, so you can get your proteins in one shape, break some hydrogen bonds, get a second shape. Now, what the crud are you doing? Why do we want to change the shape of proteins? Well, guess what? When you move your arm, guess what you're doing? You're changing the shape of the proteins in your muscle so that your arm can move. When your heart is beating, guess what? You're changing the shape of proteins to make that heart beat. We also have enzymes, which are biological catalysts. What's a catalyst?
Speeds up the reaction.
It can speed up a reaction, okay? And catalysts, our enzymes, are always going to be tertiary globs. And when this protein is in one shape, the enzyme is off. Break a couple of hydrogen bonds, we get a new shape, turns the enzyme on. Put those hydrogen bonds back, turn the enzyme off. So if we change the shape of an enzyme, we can turn it on and off. So the shape changing is super, super important. And we get this shape changing by adding a phosphate to our glob of protein. Now that phosphate comes from this molecule here. This molecule is called ATP, or adenosine triphosphate. So ATP is made out of an adenine plus a ribose sugar and 3 of our PO4 or phosphate groups. And you end up with adenosine triphosphate. We can take one of these phosphates off, add it to a protein, break some hydrogen bonds, get a shape change. If we take one of the phosphates off, we now call this adenosine diphosphate. We can borrow another phosphate, and then we have adenosine monophosphate. If we borrow all 3 phosphates, we just have the ribose sugar and the adenine, which means we just have adenosine. So this ATP, what its job is to change the shape of proteins. And anytime we're getting shape changes, ATP is involved. And when it changes the shape of enzymes, it can also help speed up chemical reactions. So sometimes you'll hear people call ATP energy of the body. But really, its true job is to change the shape of proteins. It triggers the breaking of hydrogen bonds, so we get this protein shape change. Now sometimes we get into those pathology situations where things are going to go wrong in the body. And sometimes when things go wrong in the body, we don't just get your normal shape change, or what we might call a change in conformation, a change in configuration. Those terms all mean shape change. That's totally normal. Instead, sometimes we get a shape change that's bad. And this bad shape change is what we call denaturing a protein. If a protein changes shape so much that it becomes denatured, what that means is the protein no longer can do its job, it can no longer function. So basically what happens is, let's say we've got this glob of protein and we break more hydrogen bonds than we're ever supposed to break. This glob of protein can basically fall apart. And when it falls apart, we call that denaturing the protein. And by falling apart, it no longer does its job anymore. It can no longer function. It's no longer hemoglobin, or it's no longer an enzyme. It's no longer a protein. It's just basically junk. It's cellular trash. And it's just torn apart. So to denature a protein means we break way too many hydrogen bonds and our protein loses its function. Now there's two basic ways to do this in the human body. One is to become too acidic. Remember that 7.35 to 7.45? And I told you if your patient drops to 7.2, they're very sick. The reason they're sick is that lower pH is causing their proteins to fall apart. We're talking proteins in the brain, proteins in the heart, proteins in the intestines, proteins everywhere are denaturing. They get to 6.8 and too many proteins have denatured and they die. The other thing that can happen is your temperature goes up. You get a fever. And fevers denature proteins. That's why they're dangerous. Now, how hot do I have to get before my proteins start to denature? Okay, so approximately for an adult, 40 degrees centigrade, which is about 104 degrees Fahrenheit, you get to that. Did I do that right? I can't remember all of a sudden. Yeah, okay. All of a sudden it looked bad. So you get to about 104 and your proteins will begin to denature. You don't want a fever that high.